Linked genes. Genes are said to be linked if they are placed on the same chromosome and if they are very close to each other. Usually linked genes, they inherit together. Linkage is defined as failure of the segregation of the chromosomes during the process of meiosis. Closer the two genes will be, there will be more chances that there is no crossover and the genes, they will inherit together from the parents to the offsprings. The crossing over can be measured in the terms of units. 1% crossover means that 1 map unit. 1 map unit means the chromosomes, they are closely linked, they are not far away from each other. They will inherit as a unit from the parents to the offsprings. But on the other hand, if we say that the recombinant frequency is 12%, it means they are far away from each other 12 map units. 12 map units mean they are far from each other. There is too much distance between these two type of the genes. So there is possibility that they will have a cross with each other. Crossover frequency is measured in terms of map units or map distance. We can see here that how linkage and linked genes can be understood. Here is the example of chromosome 15. In case of chromosome 15, this is the first copy of one of the homologous chromosome. This is the second copy of the second homologous chromosome, because chromosomes, they are in homologous forms. The sequence of the genes which are present on the first copy of the chromosome 15 is A, B, C and D, while the sequence of the genes or the placement of the genes on the second copy of the homologous is that can be written as capital A, B and C, D. So when they are separated during the process of meiosis, what we will say that what is linkage? One set during the anaphase of meiosis, one set if receives A, B, C, D copy of the chromosome 15, while the second set receives A, B, C, D, it means that there is no crossover between these genes or between these alphabets. Here A can be considered gene 1, B gene 2, C gene 3 and D gene 4. If the sequence is same that was present on the sequence of the chromosome which were present in the parents, if this sequence is not changed, they are in the same sequence A, B, C, D, then we will say that the genes, they are linked together and they are not separated from each other and there is no crossover. Such a genes, they are called as linked genes. To further understand the phenomena, what is the meaning of linked and recombinant? In case of recombinant, here we can see that it was AB that was exchanged with the chromatid of the second homologous. AB, it has become C and D that was on the other chromatid of the other homologous chromosome. Same is here. But in case of that, what will be the linked genes? If the same sequence comes in the offsprings or in the daughter cells, then we will say that the genes, they are linked. If they exchange the parts, then we will say that this is the recombinant, and if they do not change the parts, then they are called as linked genes. Linked genes, they inherit in the form of unit or in the form of blocks. This phenomena is particularly used for the identification of the many disease genes in case of human, where a linkage analysis is used to identify certain regions where there is a location of a gene that is causing a particular phenotype or is causing a mutation that is responsible for a phenotype.